lipids already, you'll remember, when we were talking about different types of lipids in chapter 3. And the phospholipids were kind of bipolar, right? There was part of them that was hydrophilic and part of them that was hydrophobic. So all phospholipids were like our triglycerides, right? They had a glycerol molecule that was attached to 2 fatty acid chains. But instead of it being attached to a 3rd fatty acid chain, we attached it to a phosphate. So simply, we could draw a phospholipid like this, with a hydrophilic head, and then 2 hydrophobic tails. These are the fatty acid chains, and then that hydrophilic head is the phosphate. So if you were to take a whole bunch of phospholipids and you were just to throw them into water, they would self-aggregate into something called a micelle. And that micelle would look like this. It would be a collection of hydrophilic heads and hydrophobic tails oriented in the same direction. And to protect those hydrophobic tails, or rather to hide those hydrophobic tails, there would be another layer of phospholipids beneath that top layer, so that you had a structure that looked kind of like that. So really, this fluid mosaic is a phospholipid bilayer, 2 layers. 1 layer of phospholipids, 2nd layer of phospholipids. Now, that really accomplishes our semi-permeability for the most part, right? Because we have this core that is what, hydrophobic or hydrophilic? Hydrophobic. And since it's hydrophobic, it means that it's not going to allow any polar or charged molecule to pass from outside the cell to inside the cell, or vice versa. So if there's a water molecule that we know is polar, it carries a partial positive and a partial negative charge, it's not going to be able to freely pass that plasma membrane, which is good, right? Because we want to keep the water inside our cells. We don't want to become dehydrated. Okay. Also, incorporated into the plasma membrane, there are proteins. And these proteins serve a variety of functions. And they can be like really big. Oh, I should also write that this is hydrophilic. And so is this. Okay, so we have these large proteins that can like span the whole length of the plasma membrane, like this. They can be like a channel that goes all the way across, or they can exist just on the outside of the plasma membrane. And their variety, or excuse me, their functionality varies greatly. So they can be, like I said, channel proteins and freely allow some substances to pass through the plasma membrane. One of those substances is water. So we have special pores called aquaporins that are protein channels that allow water in and allow it out. Additionally, if they exist just on the outside of the plasma membrane, they become protein tags or protein identifiers, so that when our immune system is active, it can recognize our own cells as part of our body and foreign cells as a part of a foreign body, something that we need to control and attack. They can also be enzymatic. They can also be involved in communication, intercellular communication. When one cell wants to talk to another, it can do it through these proteins that are embedded in our plasma membranes. A good example of that is if a cell gets damaged, it can tell another cell that it's been damaged so that we can start going through the cell cycle and we can start replicating new cells where that damage has occurred. That communication is accomplished via the proteins. Now the last thing that exists in our plasma membrane is cholesterol. Now remind me what type of a molecule is cholesterol? Protein, carbohydrate, lipid, nucleic acid. It's a lipid. So if it exists inside the plasma membrane, where is it going to exist? Is it going to exist close to the hydrophilic heads or in the hydrophobic tails? Hydrophobic tails, yeah. Because it's a fat, it's a hydrophobic substance. Those cholesterol molecules are all going to be located in between our phospholipid tails, like that. Now, cholesterol serves an important function. It's like a buffer. Remember, a buffer is a substance that mitigates or moderates changes in something. We've talked about buffers in the context of pH, something that's going to absorb hydronium ion or give off hydroxide or something like that. So the pH doesn't change very much. But cholesterols are really a buffer against changes in temperature. We don't want our cell membranes to get too cold, because as soon as these proteins stop moving around, and as soon as those phospholipids stop moving around, the cell dies, loses its functionality. Additionally, if it gets too hot, then those phospholipids can actually separate from each other, and the cell can break apart, which is called lysis. It can actually lyse. So when our cell membranes start getting too cold, that cholesterol inhibits two molecules from becoming frozen together to some extent. And when it starts getting too hot, it holds molecules together. So if we were to comprehensively describe the structure of the plasma membrane, it is a fluid mosaic of a phospholipid bilayer that includes proteins, phospholipids, and cholesterols. And the function, yes, please, Linda.
Can you say that one more time?
You betcha. So the plasma membrane is a fluid mosaic of phospholipid bilayer that includes proteins, phospholipids, and cholesterols.
Thank you.
Yeah. And the functionality, of course, is semi-permeability. Okay, questions about the plasma membrane? Okay. So now I think what we're going to do is talk about the difference between prokaryotic and eukaryotic organisms. Oh, no, not yet. We're going to talk about proteins in the plasma membrane. And the proteins have a varied function, right? They can be protein channels that facilitate the transport of specific compounds across the plasma membrane. They can be enzymatic. They can help catalyze specific reactions. They can receive communications, things like that. So the surface proteins, and there is a difference between where specific proteins are located on the plasma membrane. There are some, like I said, that exist just on the outside, those surface proteins. And those are primarily for marking, for individual identification. Interestingly, these proteins, which are large and somewhat complex, can have functional groups that are either carbohydrate or lipid. So there's this network of goo that exists between cells. It's called the intercellular matrix. And it consists of long strands of proteins connected to long strands of carbohydrates and lipids. An important compound in the intercellular matrix is collagen. So collagen, I think most of us are familiar with. People can get collagen injections to help make their faces look less saggy. But that's one of those proteins, surface proteins, that's connected to a long strand of like carbohydrates. And then we can have these transmembrane proteins that really facilitate the transport of substances from one side to the other side of the membrane. Now, that transport can either be passive or it can be active. And we'll talk about the difference between active and passive transport a little bit later. But in essence, what it means is, if it's a passive transport, things follow their diffusion gradient. They follow a high concentration to a low concentration gradient. They pass freely from one side to the other without us having to expend any energy. And then there can be active transport. And active transport occurs where we already have a high concentration of something inside the cell, but we want a higher concentration. So we actually invest some molecular energy into pulling something from one side of the membrane into the other. Now, these transmembrane proteins have a really interesting structure because it seems possible, right, just from this drawing, for this protein to just pop right out. It could just slip right out. But in reality, that doesn't happen because the part of the membrane, the part of the protein that is embedded internally in the membrane this part is hydrophobic, like our hydrophobic tails of our phospholipids. And this part up here that's interacting with the phosphate heads is hydrophilic. And so this hydrophobic core and hydrophilic top and bottom prevent that transmembrane protein from just popping out one side or the other, holds it in place.
I have a question. So
Okay.
that's the protein chip.
Yeah.
Okay.
Sorry, I should have labeled that.
No, you're good. I just wanted to make sure.
Okay, so this is a diagram of our plasma membrane, and you'll notice that it's largely phospholipids, that's what all these little white things are, and incorporated into the phospholipids are these cholesterol molecules. And then those big purple things are either transmembrane or surface membrane proteins. Okay, so now we're going to talk about the difference between pro and eukaryotic cells. Generally, when this lecture is given from other textbooks, you talk about the difference between pro and eukaryotic cells before you talk about the plasma membrane. But I think the way that, or the reason that our textbook has it structured this way is because all cells, whether they are pro or eukaryotic, have a plasma membrane. It's a unifying structure for all cells. Prokaryotes are incredibly simple organisms. We think that these were the first types of organisms to evolve. And they're defined as any organism that lacks internally membrane-bound organelles. So things like a nucleus, it doesn't have, or Golgi apparatus, or endoplasmic reticulum. It doesn't have any membrane on the inside of its cell, just that surface protein. And examples of prokaryotes are the bacteria and the archaea. So most of us are familiar with the bacteria, right? Things like E. coli and Streptococcus and all that kind of stuff. Archaea are defined as extremophiles. They live in incredibly extreme environments. So thermal hot springs. They live in very acidic environments like the guts of some ungulates like sheep and cows and stuff like that. There are methanogens that live in very methane-rich environments. The point is they're really simple, generally unicellular, and pretty closely related to bacteria. So what we're going to do is we're going to talk about prokaryotes from the outside structures and move our way into the inside structures. The outermost part of a prokaryote is called the capsule. And this is a protective layer that consists of polysaccharides. It's kind of like the cell wall of a plant. So you'll remember that the cell walls of plants consist of cellulose, which is a polysaccharide. Prokaryotes have something akin to that. So they have a capsule, then inside the capsule they have a cell wall, and that cell wall can either be composed of carbohydrates and proteins or proteins. The structure really varies. So hopefully at some point y'all will take microbiology, and you're going to be testing for the difference between gram-positive and gram-negative bacteria. The presence and absence of these structures are really what determines gram-positive or gram-negative. And that's an important distinction because really gram-positive and gram-negative, the approach to controlling gram-positive and gram-negative bacteria is very, very different from an antibiotic standpoint. Then inside the cell wall, so we go this capsule cell wall, we have the plasma membrane. And as you know, the plasma membrane is a fluid mosaic of phospholipid bilayer that consists of phospholipids, proteins, and cholesterols. So that's the stuff that surrounds prokaryotic cells. Inside the plasma membrane, there is what's called a nucleoid region. They don't have an organized nucleus like we do. So our nucleus is just a glob of membrane that holds on to our DNA. Their DNA is just kind of loose inside their cells, but their DNA kind of congregates in one specific area, and that specific area is referred to as the nucleoid region. Their DNA is really interesting. Ours is organized in linear chromosomes. So if you were to take all of our DNA, right, and to line it all up, each cell would have like 2 meters, a little over 6 feet of DNA in it, which is a really long molecule. And so to make that molecule a little bit more manageable, we've taken that 2 meters and chopped it up into 46 bits. But those 46 bits are all linear. They're not connected on the ends. However, prokaryotic DNA is circular, and that circular DNA is called a plasmid. In addition to DNA in that nucleoid region, they have stuff that's able to read and manufacture proteins from the DNA. And those structures are called ribosomes. Ribosomes are internal organelles, but they don't consist of any membrane. They only consist of the RNA molecule, which is another one of our nucleic acids, and some protein. Basically what happens is